Chemistry plays a major role in helping us understand biology. In this video, we'll review basic atomic structure and how electrons are arranged. In later videos in this series, we'll use this foundation to explain bonding, electronegativity, polarity, and why these ideas matter for cells. In biology, we usually use a simplified picture of atoms called the Bohr, or planetary model. It isn't perfectly accurate, but it works well for understanding basic chemical behavior. At the center of every atom is the nucleus. The nucleus contains protons and neutrons. Protons have a positive charge. Neutrons have no charge. Both have the same mass, which we measure in atomic mass units, or AMU. You may also hear this called a Dalton. The number of protons in the nucleus defines what element an atom is. For example, carbon always has 6 protons. If an atom had 7 protons, it would be nitrogen instead. Neutrons can vary in number. Atoms of the same element that differ in neutron number are called isotopes. Because the protons stay the same, isotopes behave chemically like the same element. Carbon-12, carbon-13, and carbon-14 are all carbon, because they each have 6 protons. They just contain different numbers of neutrons. Surrounding the nucleus are electrons. Electrons have a negative charge, and their mass is so small that we usually ignore it in biology. Electrons occupy regions called shells around the nucleus. The simple rule that matters here is that electrons fill shells from the inside outward and are removed from the outermost shell first. Unless we're told otherwise, we assume atoms are neutral. This means the number of electrons equals the number of protons. So oxygen, which has 8 protons, also has 8 electrons. For now, we'll focus on just the first 2 shells. The first electron shell of any atom can hold up to 2 electrons. The second shell can hold up to 8. In oxygen, of the 8 electrons required to make the atom neutral, 2 are put in the innermost shell, so only 6 are left for the outermost shell. Whichever shell is the outermost shell is called the valence shell, and the electrons in the valence shell are called valence electrons. When we draw valence electrons, we place them one at a time around the shell before pairing them. This helps to see which electrons are paired and which are unpaired. In oxygen, 4 of the valence electrons are paired, and 2 are unpaired. The number of unpaired outer electrons is called the valence number. As you can see, oxygen's valence number is 2. This matters because the valence number usually tells us how many covalent bonds an atom will form. So oxygen will form 2 covalent bonds. To summarize, the number of protons defines the element. Neutrons create isotopes. Electrons fill shells from the inside out, and unpaired electrons in the outer shell help determine bonding. In the next video, we'll look at how atoms interact with one another.